Good morning, wherever you are, or good afternoon, wherever you are. My name is Jesus Ramirez Valles. I'm professor of medicine here and also the chief of division of prevention science. And it's my great pleasure to host today's town hall on all of us. We have two wonderful speakers. The original title of this is the NIH All of Us Research Program, and our intent is to promote researcher engagement here at UCSF. I won't tell you a lot about what All of Us Research Program is, because that's the purpose of this conversation with our speakers, but I can tell you it's one of those wonderful, ambitious efforts to gather and collect health data from about 1 million people of us here in the United States that hopefully will accelerate research that may improve our individual and our public health. I have the honor to introduce two people that I can call my colleagues and my friends. First is Dr. Karriem Watson, the Chief Engagement Officer of the NIH All of Us Research Program, and then our own Robert Hyatt, who is also here at UCSF and is a principal investigator of the All of Us National Precision Medicine Initiative. For full disclosure, we're colleagues also because I'm in the advisory board of the All of Us Research Program. So to allow them all the time that they need to explain to us this and to promote our engagement with the research data, we're going to start with Dr. Watson, and then he will pass the microphone to Dr. Hyatt, and then we'll open it up for questions from the audience or discussion or comments. In the meantime, please feel free to make your comments or questions to the chat function in Zoom, and Rochelle and I will collect these questions and bring it to the attention of our speakers. Karriem, the microphone is yours.
Thank you so much, Jesus. Can you all hear me okay? Okay, yes, perfect, great. Thank you so much. I'm on VPN, so every now and then I have to make sure y'all can hear me okay. I'm excited to be here. Although I'm not at UCSF, as Jesus said, it feels like, you know, still a family reunion and a meeting amongst friends and colleagues and even mentors. I had the privilege of having Jesus as an advisor, a faculty member, and then also a PI for our Center for Health Disparities for one of the research centers minority institutions for U54 Center on Structural Violence, that I was honored to be one of the co-investigators on, that was led by Jesus and two other investigators. So really happy to be here today. And I'm going to talk to you, and also excited because of Dr. Hyatt's work, being one of the PIs for the All of Us Research Program. And I've had the privilege of working with All of Us since its inception, actually. I was one of the community members, as a graduate student and early stage investigator, I was one of the community members that helped inform the community engagement strategy for the All of Us Research Program. I used to be the co-chair of the Community Campus Partnerships for Health, an international organization that helped inform some of the community engagement principles. So you'll see a lot community engagement principles, CBPR language in the All of Us Research Program, even Karriem S type language in the program, because all that language went into the early onset of the program. And then I was fortunate enough to be one of the co-investigators for the Illinois Precision Medicine Consortium, which is one of the All of Us Research Program sites affiliated with the University of Illinois at Chicago, where I led the community engagement core for the program, and which is also part of the University of Chicago and Northwestern Medicine as part of that Illinois Precision Medicine Consortium. So, and excited to join the NIH in August of 2021. So I'm on my little over 2-year anniversary of having been with the NIH, and excited to be here. So thank you all. Next slide. So today I hope to highlight for you all, for some of you all who may know what the All of Us Research Program is, but to really, if you know about it, I'm gonna just, for hopefully you tell you a little bit something different, to really focus on how we're engaging communities historically underrepresented in biomedical research. Also gonna discuss the role of genomic data in underrepresented communities and what it will play in advancing health equity research. Even talk to you all a little bit about some of the things that I think the program can do directly for your amazing centers and programs that many of you all are leading or doing research within, and talk about our robust data set and how it can lead to support medical breakthroughs. Next slide. So just a brief historical moment to remind you that back in 2015, January 2015, then President Barack Obama gave a historic speech that talked the State of the Union address that really launched the Precision Medicine Initiative. And the All of Us Research Program is actually funded out of the Cures Act. And he quoted and said that tonight I'm launching a new precision medicine initiative to bring us closer to curing diseases like cancer and diabetes, and to give us all access to the personalized information we need to keep ourselves and our families healthier. Back in January 2015, this is really thinking about if we're going to think about personalized medicine, healthcare for all, how do we ensure that research and other innovative treatments can reach everyone as well? Some of the things that we know is that sometimes innovation in science can actually unintentionally cause a bigger disparity gap than we intend to, because if we're not careful, those precision medicine initiatives and innovative science can reach some populations and not all populations. And sometimes we can reach those populations who are more resourced as opposed to those populations who are historically underrepresented. Initially, the All of Us Research Program was called the Precision Medicine Initiative. There was a working group that was launched in September of 2015. And then in December 2016, the 21st Century Cures Act, enacted by Congress and signed into law, set the funds in place to support programs like the All of Us Research Program. And unlike some research programs that are asking for a particular question, trying to determine if intervention A is better than intervention B, or I'm trained as a cancer disparities researcher and trained in community-based participatory research, CBPR, where my research often looked at community-based screening programs and how to identify barriers and facilitators of some populations getting screened for cancers and other health disparities diseases. All of us is looking to collect data from 1 million or more people living in the United States to accelerate research and to improve health. So unlike some research programs that are asking particular questions, all of us is setting out to collect data from 1 million or more people and make that data available to researchers and even community and citizen scientists to be able to interrogate that data and address health disparities. Next slide. So where are we today? And Dr. Hyatt, I hope if you want to talk about the numbers too for what you all are doing and the California Precision Medicine Initiative, but I'll just briefly tell you a little bit about where we are in terms of other genomic programs. So we're over 700,000 participants have signed up to join the All of Us Research Program. And of those 700,000 participants, we actually have data from over 488,000 who have completed the initial steps of the program. And that data gives us over 504,000 biosamples with access to over almost 400,000 electronic health records. Actually, I think as of this weekend, we actually crossed the 400,000 mark for electronic health records. So you think about what that means from a longitudinal perspective for access to study data. You think about what it looks like for someone to have been, we are very fortunate right now that HIV and AIDS is now a chronic condition. So imagine having EHR data from someone who was diagnosed 20 years ago, being able to follow them, look at their renal function, look at their renal labs and other things over time to see how things may have changed in response to antiretroviral treatment and other things. Those are some of the things that we're really excited about that you will have access to in the All of Us Research database. Another thing that makes our program innovative, I want to draw your attention to the other genomic studies pie chart on the bottom. Historically, genomic studies that collect this type of data we're collecting in the All of Us Research Program is historically over 90% white or European descent. And what we do know is that lack of diversity leads often to a lack of generalizability to other populations. You think about the presence of African-American or Afro-Caribbean groups in larger genomic studies, they're less than 1%. Think about Asian Americans and Hispanic and Latino groups, also less than 1%. But in the All of Us Research Program, our Black or African American population is it's at 17.2%. Hispanic and Latino participants are at 16%. Middle Eastern, Northern African, Native Hawaiian, or does not fully describe me, about 2.7%. White, we're at 52.2%. And even soon, we're actually in the process of conducting tribal consultations with American Indian Alaska Native tribal leaders. After we can continue conducting those tribal consultations, we will actually have a plan to also release data on American Indian Alaska Native populations to further release data on populations who have been historically underrepresented in biomedical research. Next slide. So prioritizing intersectionality and social determinants of health. As I was saying earlier, so I was funded by the National Institute of Minority Health Disparity, the NIMHD, as a cancer researcher with a colleague of mine, former colleague of mine at UIC, Dr. Sage Kim. And together, Sage and I got an NIH R01 looking at lung cancer disparities in African-American men. But I remember working with Sage and working with Dr. Kim to get that preliminary data for tobacco use amongst African-American men. It took us about 3 years to really get the preliminary data we needed that could be powered up to address the intersectionality that we needed, because a lot of the large secondary databases did not have intersectionality data on African-American men. Databases like NHANES and other databases that were great, they didn't have that data that we needed. So imagine the fact that All of Us research program actually collects data from what we call UBR categories, which includes race and ethnicity, with 47% of our participants being from an underrepresented racial or ethnic minority group. 26.1% of our participants are greater than 65 at the age of consent. So that's 1 in 4. And as we think about the aging of America, this data becomes really important as well. 25% of our participants have income less than $25,000. About 18% of our participants are living with a disability. 10% are sexual and gender minorities. Sexual orientation, not straight, about 10%. Educational attainment, about 9.1% of our participants have less than a GED. 7.4% of our participants live in a rural area. And when you think about what this means in terms of research, this intersectionality. So I want to now just draw your attention to, if you are on the call or in the town hall and you do HIV or AIDS research or prevention research, and you want to say, Karriem, you know, how many participants in your program have been identified as living with HIV or living with AIDS? Our EHR data from over 254,000 people show that we have about 5,500 participants who have self-identified from their EHR have noted that they are living with HIV or living with AIDS. And of those that are asymptomatic, we have over 4,000. So imagine what that data set can do when you start thinking about intersectionality. And then if we go further into the data and start looking at electronic health records, labs, and other measurements, we see that over 45,000 we have HIV tests on over 45,000 of our participants. And we think about other things, detection tests by immunoassay technique, over 6,700. You may say, well, these numbers differ from those that are living with HIV in the EHR, but the point that we're trying to make here is the amazing intersectionality data that we have. Next slide. So other types of data that makes our program unique. In addition to the genomic data, we collect blood and saliva samples to get at the genomic data, to get at whole genome sequences and look at structural variants and genotyping arrays. But we also collect physical measurements such as height, weight, blood pressure, and other things we can also get from physical measurements, but also get from the EHR. We also collect survey responses. When you think about the survey response, we have a social terms of health survey that was developed by some colleagues at Mass General and other members that are part of the social terms of health working group. That SDOH survey was actually completed by over 200,000 participants. And so if you couple that data that we have from SDOH, I recently talked to a cancer disparities researcher at Morehouse School of Medicine who was looking at vitamin D deficiency in African-American men with aggressive prostate cancer. And he wanted to know, Karriem, is your database able to show me African-American men that have been diagnosed with prostate cancer? Yes. Could we actually get access to vitamin D levels in those African-American men? If they had it in the EHR through a lab value, yes. Could you actually get access to some social demographic questions? You could. And then he took it a step further and said, what if I wanted to know of those that are living with aggressive prostate cancer that had low access to vitamin D, are they living in a food desert or a rural community? The answer to all that was yes, he could get access to that information. So now we have researchers even thinking about how this data set can be used to support K award applications or other NIH R01 and R21 type applications. So hopefully that what you'll see is that from this wide array of the different type of data that we collect, the different types of research questions that we're able to ask from the All of Us research database, even questions from Fitbit, from wearable device. I'm excited that we now have sleep data that's available from our population. And when I came into the program as a chief engagement officer, I was really, Jesus, I think you'll be really happy. I was really vocal. I had all my mentors in my ear saying, Fitbit data. Well, this data is only going to be available to a certain, those that have Fitbits are only be a population that are well-resourced. Well, the program heard me, and as a result, they passed out Fitbits randomly and selectively to populations who ever wanted to receive them. So if you had signed up for our program, you got an email and said, if you want to join our study and provide information via Fitbit, You can sign up and get a free Fitbit that was sent out to you. I was really proud of the program for doing that because that meant that populations who historically are not able to provide wearable data were able to provide that data. Next slide. So this is our scientific priorities roadmap that was made by Dr. Jeff Ginsburg and his team. Dr. Ginsburg is our chief of our Division of Medical Scientific Research. This simply shows that at the workbench and data level, the type of information, the new methods and toolbox and phenotype expression that we're looking for through EHRs, surveys, physical measures, and digital tech technologies, and genome assays, thinking about what that does in informing environmental health inequity, genetics and biology and lifestyles, and modifiers that may exist, ultimately leading to research that can be translational at the government, industry, and health systems, and even the payer and policy level. So really excited about our ability to inform individual and population health in the All of Us research program. Next slide. So this is what we call our data roadmap. So this shows you what's available at the end of 2022, what we have available now, and then what we'll have available in 2023 and beyond. So as you think about questions that you or your students or your postdocs may want to ask that utilizing our database, one of the big questions I get asked as a cancer disparities researcher is about environmental exposures. And even when we think about, I think about being in the Chicagoland area where I am, the 3-digit zip code level is great, but it would not get at the data that I need, because in Chicago, for example, by 2-digit zip code, you can see 2 different life expectancies. You can see a change, and this is 20-year life difference just by a 2 zip code change level in the Chicagoland area. 20 years. You can take one of our public transportation lines going from one community to the next, from the south side to the north side, and that life expectancy can change by over 20 years. So that 3-digit zip code level, it's great, but I'm excited that we'll be able to get to that 5-digit zip code level in our control tier. And I'll tell you more what we mean by that control tier level. And we also launched ancillary studies like Exploring the Mind, which is looking at mental health and well-being surveys, and also a newly study that was launched, Nutrition for Precision Health. Dr. Hyatt, is UCSF part of the NPH study?
Yes.
Okay, awesome. Thank you. Next slide. This is just an example of some publications that we have. So publications and research findings. To date, we have over 7,000 registered researchers with over 200 plus publications in peer-reviewed journals and over 700 citations to mark the All of Us, using the 2019 All of Us marker paper. And just some examples of papers that have addressed health disparities that many of you all may be interested in. We have one paper that was led by Yang ETL looking at computational phenotyping with the All of Us research program, identifying underrepresented people with HIV or at risk of HIV. Another study looked at protocol for developing a personalized prediction model for viral suppression amongst underrepresented populations in the context of the COVID-19 pandemic. Another study that was done by one of our National Community Engagement Partner awardees, Dr. Juno Maliver, and Mitchell Lunn at Stanford University, looked at the prevalence of the 12 common health conditions in sexual and gender minority participants in the All of Us research program. And really excited that I'll talk about who our national engagement partners are and how they've helped lead some of this research. Next slide. This is an example of our community engagement partners. We are really excited that we have over 17 national community engagement partners. And when you think about populations who are historically underrepresented in research, you will see that we probably have a representation of one of those populations as one of our national community engagement partners. For example, the Asian Health Coalition has been with us for over 5 years. And one of the things that we're excited about that's going to be coming out in our data is historically health disparities for Asian Americans has been aggregated. And a lot of those health disparities have been hidden, for example. In our new data set, you'll be able to disaggregate data out by ethnicity for different populations. So you'll be able to see someone may self-identify as Asian American, but if they also self-identify as being Vietnamese or Chinese or Japanese, for example, you'll be able to see what health disparities that may be hidden or that may exist within those sub-ethnic populations. And we're really excited about that and what that will mean. Also, the National Alliance for Hispanic Health has been a huge partner, an important partner for us. Our program actually launched in both English and Spanish. And that's been one of the successful reasons for why we have been able to enroll a large Hispanic and Latino population, even Spanish speaking as well. Next slide. So this is I'm getting ready to finish up and turn the floor over to Dr. Hyatt so we can have plenty of time for discussion. But this is an example of an adapted socio-ecological model. One of the things we wanted to highlight is that we consider engagement a science. And so what we've done is adapted a way in which we can conduct engagement and outreach, thinking about how do we reach out to populations who are historically underrepresented, and how do we foster trust? How do we build relationships? How do we develop materials that can provide information, not just about the All of Us research program, but about research in general? How do we promote education and access, activities that foster and build upon each other in terms to provide resources, information, and the ability for people to make an informed decision and about participating in research? I was recently in the south suburbs of Chicago at an institution called Governor State, which is a small minority serving institution in our south suburbs. And I had to talk about, I was on a panel with the grandson of Henrietta Lacks. I was on the panel with Alfred Lacks Carter. And he talked about that he still has to tell people that when his grandmother, when that violation happened against Henrietta Lacks, the IRB was not in existence. Informed consent documents were not in existence. So still today, we have a lot of education to do for communities to let them know, A, to acknowledge that these historical things and these atrocities have happened, but B, to also talk about what things are in place. We also talk about engaging to enroll and retain, then talk about participants as partners. We have a full integration of our participants in several forms of governance. And then how do we mobilize knowledge in our program as well through both disseminations and conferences? Next slide. Here's an example of how we operationalize our community engagement framework. Just in 2022, the Asian Health Coalition reached over 158,000 Asian American, Native Hawaiian, Pacific Islanders across the nation by hosting 391 virtual and in-person events conducted by 22 sub-partners that they have. The National Alliance for Hispanic Health collaborated with our mobile engagement asset, which is a mobile van, a mobile home that goes across the US to engage populations that may be in rural communities or communities that don't have access to an enrollment site like UCSF, so that they can learn about the program and even sign up if they want. Just in a short 3-month period, the National Alliance for Hispanic Health was able to create 211 accounts just in 3 months. And then Pixis Partners, one of our larger engagement partners, actually created a network of CPGI partners that has over 151 smaller community partners that's really able to address community engagement. And then we are one of the few government programs that actually has participants as partners. We have a participant ambassador program with over 35 participant ambassadors that sit in steering committees, executive panels, and even our IRB. One of our colleagues at Hunter College and Northwell Health, Dr. Liz Cohn, actually conducted 2 data sandbox with community members, where she takes community members to our database, shows them what data is in there, and asks them what things in this database are important to you. Next slide. This is an example of community convenings. We're getting ready to host a community convening for Hispanic Heritage Month in San Diego, California on Saturday, where we'll have over 130 community members convening in San Diego to talk about return of value and what precision medicine means for certain populations. This is an example of a convening that we hosted in Tuskegee, Alabama. That picture on the right there, you'll see, that's a historic picture. The lady in the red is actually Lily Head. Miss Lily Head is a descendant of one of the participants from the United States Public Health Service study of untreated syphilis in the African-American male. Some people call that study the Tuskegee experiment. When we were in Tuskegee, Alabama, they actually asked us that we no longer call that study the Tuskegee experiment. Tuskegee said we did not ask for that study to be conducted, and it brings such a negative stigma to Tuskegee by calling it the Tuskegee experiment. But it was the first time that descendants of Henrietta Lacks and descendants from that study were on the same panel. And one of the quotes that we have is that we're moving promise to progress. Family members of victims of well-known medical transgressions speak with health leaders on working to regain trust. Next slide. And I'll see some questions in the chat and we'll come to those. This is another example of returning value. And just a personal story here is that I've dealt with a lot of health disparities within our family with cancer disparities. My birth mother died of breast cancer when I was 6, which really led to me wanting to do cancer disparities research. My father and my oldest brother died of colorectal cancer before the age of 60. So you can imagine, as both a cancer researcher and as someone who's been impacted by cancer at an early age, I was concerned that we may have something like Lynch syndrome or other things in our family history. Thankfully, I was tested and we do not have those things. But when I got my return, because I'm also a participant in the program, when I got my information returned back to me from my medicine and DNA, I actually found that I had a gene that could affect how I metabolize certain blood thinners. When I began to have conversations with my primary care providers and other family members were shocked and asking me questions like, well, is this the reason why our aunt, who was on all her blood pressure medication, who took her blood thinner, but still had a stroke, still had a follow-up stroke to that? I don't know if that's the case, but the fact that I now know that in the unfortunate event, if I were to have some type of cerebral vascular event, that my doctor should really look at the type of blood thinner they put me on based on my medicine and my DNA information that I know now. But I also now know that I'm not at risk for colon cancer based on a familial genetic predisposition like Lynch syndrome or something else. But I still want to know what's going on. And that gets at that intersectionality, not just the genes, but everything else that we have going on. Next slide. This is our in our research engagement office, in Division of Engagement and Outreach. We also are excited that we engage researchers as from all the way from high school all the way to community and citizen scientists. And we would love to talk to you later about ways that you can incorporate the community and how you utilize the workbench. The Ignite team is really excited about doing that work as well. Next slide. This is just what data is available in the public tier, the registered tier, and the control tier. But I'll save this information for the Q&A because I want to make sure Dr. Hyatt has time for his talk and that we have plenty of time for Q&A at the end. Next slide. And this is some research coming out of our program, looking at anticipated findings, looking at research, looking at anticoagulants in our data set that I talked about, and I'll put links to these studies in in the link. Next slide. Another study is looking at using the All of Us data set to explore risk factors for comorbid depression in patients with HIV. This is a project that's coming out of the University of Puerto Rico. Next slide. And this is a case study that I'll save for the Q&A, but the purpose of this case study that we'll get to in the Q&A is just tell you what's possible and how you can think about how you can utilize the All of Us data set for a lot of intersectionality data. So with that, I'm going to ask Angelique to go to our closing slide. Thank you all for your time, and I'm going to turn the floor over to Dr. Hyatt now.
What I'll try to do is to give you a little local perspective from UCSF. Turns out Dr. Watson and I probably know each other for 5 years or so, because when he was at University of Illinois in Chicago at the cancer center there, I was one of the advisors that came and helped them conceptualize their community engagement office. And I worked with Robert Wynn then, and I continued to do that. So this is great. Also, before the talk, we were all chatting, and turns out many of us have gone through University of Michigan and Ann Arbor in our lives, so we made a bunch of nice contacts. So, like I say, Karriem covered a lot of important information, and I'll skip over some of the stuff that I have, which would be duplicative, but try to give you the sense of what this program looks like from UCSF. I'm a professor of epidemiology and biostatistics in our school, and also associate director for population science at the Cancer Center. So that's been my work experience. And all of this is one of several projects I work on. Very excited to be the PI, site PI from UCSF. So let me go through some slides here with you. So Karriem highlighted President Obama's initiation of this project. This is Francis Collins, who I know from personal experience had worked on getting a large cohort like this for many, many years, and was very supportive of the development of the All of Us research program from the NIH. And as Dr. Watson has said, the idea is to gather data from 1 million or more people to accelerate research and improve health nationwide. It isn't a hypothesis-driven study. It has a lot of different data that allow you as researchers to come up with your own ideas and own hypotheses. And we're trying to make it as rich as possible to answer a whole series of questions. Okay, so this is the key thing. So this is a really diverse group, as Dr. Watson has pointed out. It's diverse not only from race and ethnicity standpoint, but also from where people live, what they work on, their medical backgrounds. And there's no exclusion criteria for this. This is something that I want to emphasize. So people that are already afflicted with diseases can be members as well. So that means that you can do studies on individuals who already have diagnosed diabetes or heart disease or cancer, as well as those people who aren't, who you can follow along longitudinally for the development of events. And the other unique thing is the governance. So we're not just doing this as researchers, diving into the community and parachuting back out again, parachuting in and then diving back out. We're participating with the people who are joining us, and we call them our participants and our partners. We don't call them subjects. We don't call them anything but partners in the process that we're doing. And this is a national resource, as I say. So how does it work? This is how it works, not only at UCSF, but everywhere. So we are recruiting people who are 18 years old, and we look at including children, maybe next year. We're already talking with the Benioff hospitals to make that happen. The consent is online and interactive. Importantly, it asks for the electronic health record. I have to say some people balk at that and don't want to join, but it's a critical way for us to have additional information and to follow up for outcomes. And then they also have to consent to get their DNA results if they want them. They don't have to. Then we collect a lot of data, and there's a whole list of modules which are not given at the same time to think about participant burden, but they are quite diverse in what they ask, including the social determinants of health. This is a module that I personally worked on, and we're getting a publication ready now to describe the methodology. But it was led by Cheryl Clark from Harvard, but involved a bunch of us from around the country, and adds a really rich aspect to research in terms of things like food insecurity and housing insecurity and racial structural racism, transportation, other things that are part and parcel of our social fabric, which are important to study. We have the physical measurements you see there, and then we have a blood and urine sample that's collected and sent off every night to, in the case of blood, to and urine, to Mayo Clinic in Rochester, a truly fantastic accomplishment, I think, in terms of logistics and so forth. And then the data goes every night to Vanderbilt in Tennessee, where it's managed and put together and cleaned and ready for analyses. And then there's the story with the Fitbit, and there'll probably be some other types of data collected soon. So you could ask, why do we need this program? And I think Karriem mentioned one of the big reasons, and that's most of the big epidemiologic studies. And I'm an epidemiologist, and I've spent most of my career building these large cohorts, but most of them are white folks. So the Harvard cohorts, Nurses Health Study, Framingham, are all heavily white individuals. Even the multi-ethnic cohort in the West Coast has a large proportion of white individuals as well as Asians. But getting to have a representative sample is not only important for social justice and representation, but there's, epidemiologically speaking, enough individuals in these different subsets that you can actually do studies of, Chinese women above 80 who have diabetes, some other intersection of conditions, for example. Healthcare providers are interested in what we're now calling precision medicine. So they want to know what are the characteristics not only of the genome and their omes, but what about their social and behavioral context in order to understand etiology as well as therapeutic approaches. And then those of us that are in the business really are benefiting from having such a large data set that reduces the time and cost it takes to develop a cohort. Again, I've been at it for a long time. I've probably developed 4 or 5 different large cohorts, and 80% of the time is spent collecting the data, putting it together, and 20% is actually using it and developing analyses. So this is a real benefit to the community of biomedical researchers. So how does it lead to discoveries? What's going to be the process? I want to move my banner out of the way so I can see things, but I guess I can't do that easily enough. But anyway, so participants are sharing data, as I said. The data are protected. We've spent over a lot of efforts to make sure that it's as confidential as possible and as protected as possible. Then researchers are invited to study and use the data, and that's what we're doing with you today. And I'll go back to that. And then the participants get information back. So not only from the studies that are done, but their own studies. So researchers now, participants now can get omic data on various characteristics, but also their particular SNP abnormalities, which may lead to major diseases. So the 59 genetic abnormalities, which are recognized by the American Human Genetics Association, are looked at, and I think that's going to be expanded to 79 in the near future. And then the researchers, of course, share discoveries across the board. So this, unlike other cohorts, is not waiting for the total results. The data is available now, and you can do studies on what's available. And Karriem has showed you the large numbers that are already available. So this is what we're hoping to do. So we have people and health status and geography and data types, and we can incentivize what are called health provider organization networks. UCSF is one of these, to focus on underrepresented and biomedical research communities. So a lot of our efforts going into pulling people into the study, explaining what it's about, building their trust so that we get a good representation across the board. Some of that can be done through federally qualified health centers that are valuable partners in this. We actually ask direct volunteers, people who are not members of HPOs or other universities, to participate as well. And you'll see how we do that. And Karriem has told you a lot about population engagement already. I'll just say a word about privacy and security, because it's really important to a lot of people. Some people don't want to join because they just don't trust that the data will be kept secure. But in fact, it's about as secure as anything has been done in my experience previously. So there's certificates of confidentiality. The data is encrypted. Only certain people get access to it. If you are looking at genomic information, you need to be vetted through a process which has been developed, and participants themselves can actually look at data at the most elemental levels and see where they fit in the bigger picture. So lots of things that can be done, but just to highlight a couple things, we can estimate risk, standard epidemiologic undertaking. Pharmacogenetics is really important because it turns out there are some genetic predispositions to make you sensitive to certain drugs, either over or under metabolize them. So markers are being identified for pharmacogenomics. Other biological markers that come from both blood and urine that can identify environmental exposures. And solutions to health disparities, that's a bit of a too much of an abbreviation because that's huge. and we're working heavily to understand health inequities, which we observe as differences. But health inequities are disparities that are unjust, unfair, and avoidable. That's what we're after. We're using mobile health technologies, as mentioned, and the participants are part of the process. So even in our local group, we have participants that are giving us ideas and some feedback. So here's the major building blocks. This is sort of a summary of what you can look at. So there's data, there's the biobank, there's participant technology that goes into helping people understand what's there and how to use it. There's partners we have from Baylor and the Broad Institute and Color, University of Washington, that are helping us with the not only the genomics, but the counseling that has to go with it. And we have a participant center that's working with direct volunteers, healthcare provider networks, which is what we are. And I should say, in California, we have 6 different institutions, not just UCSF, but also UC Irvine, UC Davis, UC San Diego, as well as USC and Cedars in Southern California. So the 6 of us all do the same thing. We meet regularly and we work as a consortium. And then there's a communication engagement network that is critical to the whole thing, which Karriem has emphasized. So yeah, there's new things coming up all the time. One of it is the Fitbit and other exercise-related tools. Data is being linked to other sorts of data, from geospatial mapping and medical records data from claims. And as I say, the social determinants of health data has helped us already substantially. Some of the projects can be directed by consortia with participants. If they have good ideas, we can help them develop their own studies. And this year, the focus is not only on retention and recruitment, but also on getting more researchers to use the data. So again, this is a reminder, but there's a lot of data already there. Over, I think, almost 500,000, 600,000 participants at this point. And they have provided multiple surveys, electronic health records, physical measurements, genomic data is being determined, and biosamples are being collected and analyzed. So there's a lot of stuff in there. This is something that Karriem mentioned. I want to go over it again. So there's three levels of data. The public tier is one that you can just go into right now this afternoon and get these little data snapshots of what's in there. Size of the population, their general demographics, and what kind of things are available for study. If you're interested in becoming a researcher, then you can sign up and get access to more in-depth data from surveys and electronic health records and wearables and physical measurements. And then there's a more restricted tier, final tier, which gives you more data on genomics and expanded demographic stuff that's really personal and most highly protected, but also very valuable for understanding disease and its consequences. This is how it looks if you were to put an org chart around it. But the public tier is available to anyone. Registered tier available to registered users, which you can become by signing up. And then the controlled tier is available to registered users, which have institutional agreements to participate in this data. This is what it looks like at UCSF as of yesterday. Excuse me. We have recruited about 7,200 participants, and about 5,000 plus have finished all the core requirements, which means electronic health record, the physical examination, filling out all the modules. And there's a difference between being actively and passively retained. Active retained means we've recently had contact with them through a questionnaire or some other process, so we know they're out there and still there. And then the EHR gives us a chance to retain people passively. So we know 75% at least of our participants are available because their EHRs are continuing to generate data. And we have really more than 350 folks from UCSF already using the data. So this isn't a novel or a strange thing to do. The other thing that's UCSF specific is our own activities around engagement and enrollment. So this is what Dr. Watson has been doing nationally, but locally, we are working with all sorts of different community groups to let them know about the study and to try to encourage participation, to build trust, and to make this really applicable to large number of people and institutions in the Bay Area. We've worked very closely with the PrideNet from Stanford. It's run by Mitch Lund, who many of you may know from when he was here at UCSF, nephrologist. But he's running the PrideNet group and has been really critical in recruiting LGBTQ populations to the study. We went down to Stanford one day with that van and recruited, I think, 30 people in one day with their help. So just to recap here before I finish, there's a number of things which have already been done. There was a COVID survey, which generated a lot of information on all the topics you can see listed there, and as well as some mental health information. This is one of the largest collections of mental health data that exists, collected in a standardized way. And then, as I say, we're collecting now data on the social determinants of health. Sorry about my voice, but it's just the beginning. There's more wearables coming, more surveys, and more ancillary studies in the wings. So finally, the advantages of working with this data are that it's highly diverse. 80% come from communities which are underrepresented in biomedical research for one of the reasons we've listed. The dimensionality of it, you can make associations across multiple different diseases and types of participants. There's a lot of detail, granularity of participant data. It's broadly accessible, which is really novel. So not only participants, not only researchers, but participants can get into the data and be informed. And the data is available fairly quickly. The limitation on the data availability is that you have to know something about R or Python, but you can get help by doing that, And so far, there's limitations on the environmental data available, but that's coming along very shortly. So, yeah, already said this, but it's already being used, and it's available for you all to jump on board. We have workspaces, notebooks, data builders. There's a lot of help for getting involved if you have particular questions. And this is my next to the final slide. These are the members of the consortium. It's quite large. And you can see we're over here on the left. But we have groups participating from all over the country. It's really exciting to work with such a broad group of people. And these are the community groups that are involved. And as you just scan through this huge screen, you can get an idea of the wide participation, all sectors of society. So that'll do it for me. I'll get a glass of water ready here and try to answer your questions. Thank you very much.
Thank you, Robert and Karriem. This is so wonderful and at the same time overwhelming. There's so much potential on this, and I hope the audience is capturing this. I'm gonna comment quickly and then I pass to the audience. The purpose here is engagement. So those of you asking questions and looking for resources, the chat is full of links and resources, and this is being recorded too. So take advantage of it. quickly, Karriem or Robert, are we going to have RFAs through NIH to take use of this data?
Yes, I'm so happy you mentioned that. I'll put the link in, or Angela probably going to beat me to it. There's actually some R21 applications that are due November 8th. I know that may sound like a quick turnaround time, but if you have a shovel-ready proposal that you've been thinking about, the first round of those ROAs is, R21s is due on November 8th. But then there's other opportunities for what we call a researcher opportunity announcement that has even engagement awards. So all of us offers research funding from everything from community engagement all the way to using our database to develop research questions through R01s, R21s, and R03s. Thank you, Angela.
Excellent.
That link is in there. So
questions from the audience? Edwin, do I see your hand?
Yes. Although, thank you, Bob and Karriem, for answering in the chat my question. So we're part of NIMHD's Health Equity Action Network. We actually are the coordinating center for it, which is like 11 disparities research centers around. And one of the things that we definitely want to try and increase our connections with all of us. One of the possibilities is we have, as one of our aims, we work a lot with the ESIs from each of these 11 disparities research centers. And I just imagine being able to have a workshop where early stage investigators can try out and collect pilot data on potential relationships that they may be interested in using the All of Us data would be a really great skill set to have, a great resource to be connected to. And I saw that as kind of like sandbox thing that you talked about from, I think, Hunter College, et cetera. What's actually involved in that? And do you guys have much experience in organizing and helping folks who want to create these kinds of workshops and how-tos for early stage investigators who are particularly URM themselves?
100%, Edwin. Thank you. We actually worked with several NIMHD centers already. So two things. One, we do supplements to some centers. So a year ago we did administrative supplements to researchers that are funded through research centers, RCMIs, which are research center minority institutions. This year we did administrative supplements to researchers that are funded through the NCI Patchy network, which is the Partnerships to Advance Cancer Health Equity Disparity. Those are ways, but the other way that I think you could benefit most is by contacting us. We partner with Baylor College of Medicine, and we give data boot camps and data workshops. We can actually bring in our research team from Vanderbilt, Baylor College of Medicine, and we can do a workshop with your early stage investigators about what is possible. They can do live demonstrations from the workbench about what questions can ask, how to formulate those questions, what skills in R and Python are needed. If you had MS ESIs that even needed R and Python's training and support, we can also provide that as well.
Great, fantastic. Okay, so I will reach out. I will be reaching out to you in the next 2 weeks or so, just to get that conversation started. Thank you so much.
Oh, we're excited.
Questions, comments?
So Edwin,
sorry.
One of the things that I noticed when we were putting together the module on the social determinants of health is that NIMHD has also developed I think it's called the FENIX questions that deal with the same territory. And some researchers should look at the two approaches to determining the social determinants of health and see how they provide the same or different data. It's a sort of a methodologic question, but I think important as we try to find ways of measuring and tracking social determinants of health.
Yes, our network is actually extensively relying on the FENIX for its own. One of the requirements from NIMHD for this network was a set of common data elements that all of the 30 intervention trials and hopefully the pilot studies have to use as well. So looking at the correspondence between what's in All of Us and what's in this network, I think would be really useful. And I can task our folks to do that.
Yeah, that sounds like you're well positioned to do that. That would be great great service.
Okay, sorry to interrupt you, but I have Dr. Koster raising her hand.
Hi. Oh my goodness. Thank you so much, Karriem and Robert. This is beyond exciting. My question relates to the inclusion of younger people. And I think, Karriem, you may have mentioned that you're hoping to enroll children. And I guess I just wondered if you could talk a little bit about, might there be a way to stage it so that, I guess I'm just thinking it would be complicated to enroll children, but could you?
Oh, it's very complicated.
16 and older? I don't know.
No, we're gonna enroll birth to 17,
Birth to
but
17, okay.
but it's gonna be staged out. We just hired Dr. Sarah Vendries, who is an internationally recognized pediatrician. She comes from Vanderbilt and she's built a lot of pharmacogenomic data sets. But to let you know how staged out this is gonna be, we're actually gonna only start with 5, what we're calling microsites that will launch, that will show us what's feasible, what does it take. We're even doing a community engagement approach where we're hosting community engagement listening sessions across the country with parents and guardians to talk about what that will look like. That's happening right now. Then information we get from those listening sessions already has told us the need to phase this out. So from now to December, we have 5 sites that you can only enroll if you are part of the consortium already. And if you are a parent or guardian, your child, if you've one from those 5 sites, can enroll. But that's going to be such a small cohort, looking at a very intentional way. And you have to be birthed to 4 years old for the first cohort. Then we'll phase it out from there. But I can give you more detail about that. Now we're running out of time.
I'll just say locally, 2 of the 6 sites are involved, but they're just being asked to recruit, I think, 1 or 2 kids to start out with, to see how it goes. And the other thing to say is that we've had a lot of enthusiasm from the Benioff Children's Hospitals in San Francisco and Oakland to participate in this.
Excellent. Thank you. Thank you. This is so wonderful. And I really appreciate Dr. Watson and Hyatt. I know you are super busy. Thank you for taking this opportunity to engage with us. And the audience, please take advantage of this. We have here in-house Dr. Hyatt, and then we have Dr. Watson, who's just leading wonderfully the community engagement across the country. And then you have me, if you want to say something to the advisory board of all of us. And this is for today. Thank you, everyone, for your participation and for completing the survey. Thank you, Robert. Thank you, Karriem.
Okay, adios.
Adios. Ciao.